Hi guys, this video is going to be over the cell cycle and mitosis, and I really always enjoy starting it off with this quote. Biology is the only subject in which multiplication is the same as division, because that's exactly what happens when your body divides mitotically. You'll see. Here we go. Okay, so your cell cycle is all about cell growth and then eventually cell division. So we'll step through it. We will cover regulation of the cell cycle and all the cancer stuff in depth later in a different video. So the cell that every living thing, person, whatever, starts off as is about the size of a period at the end of the sentence. So just for reference, this is how big you were at some point, like when your life initially started. That's how big you were. Look at you now. How did it happen? Series of divisions. Over and over and over again. One cell became two. Two cells, four. Four cells, 8. And it just kept on happening and happening and happening until you became a multicellular organism. So why would a cell even divide? Excuse me. Sorry. Only ever get sick when I have to make videos for y'all. Anyway, so cells divide for 3 main reasons. 1, so that you can reproduce. 2, for growth. And 3, for repair and renewal. So a lot of reproduction, especially on a basic lower level organismal, I'm so sorry, excuse me. Like I was saying, a lot of reproduction for basic lower level organisms is going to be asexual, where one-celled organisms split themselves through a process called binary fission, and they make 2 of themselves. It's almost like you're cloning. In terms of growth, you're adding more sections, more regions to something that already exists. So from one little tiny fertilized egg to a multicellular organism. And then for repair and renewal, to replace cells that die. Unfortunately, cells have a fairly short lifespan, especially when we're talking about human cells. Considering that a person can live 80, 90, sometimes even 100 and plus years, your cells aren't going to really stick around. At least most of them won't stick around for that long. So you're continuously replacing the ones that have worn out or have already died. Then there's also just normal wear and tear. Between the ages of about 2 and maybe about 10, I was a gigantic scab. I always had a Band-Aid on some part of my body because I was falling down, scraping myself, cutting myself, running into something, had a bruise, and I think we all did that when we were little kids. Even as an adult, occasionally, you're gonna not take such good care of yourself or not be as careful as you should be, and you'll have to replace some cells. So what are we moving along? What are we passing along when we make new cells? Well, when we make daughter cells, we are taking our genetic material and we are making an exact copy of that genetic material. And that process is called mitosis. When we go ahead and we copy all of the organelles and all of the cytoplasm and the membrane and all the enzymes, that's also a part of mitosis. But then eventually the cell is going to split itself and we're going to form 2 new cells, and that process, the actual splitting, is through cytokinesis. So, just to give you a reference, excuse me, sorry about that. So, to give you a reference, this is a cell, one cell. Everything stained in green is part of the cytoskeleton's fibers, whereas all the orange parts are your chromosomes. Now, this is not a human cell. It's actually a cell from a kangaroo, oh boy, rat. I actually had one of these. Falcor. That's him. He died. It's a sad story. Tell you later. Actually, it wasn't that sad of a story. He lived as long as he possibly could, and then he died. He was a great pet, though. So, in terms of the cell cycle itself, it's broken up into 3 or 4 main regions. One of those small sections is where mitosis itself takes place. So this actually shouldn't say overview of cell cycle. It should say overview of mitosis. Sorry, guys. I don't know. Perfectly fine before I started making the video. Anyway, and we'll step through this in a little bit. So we're going to move on from this slide. So let's talk about the cell cycle. This is the actual cell cycle. We can really take the cell cycle and split it up into two sections. The biggest section is called interphase. And interphase is just your everyday life. We're just living and growing and doing all the stuff that we have to do to be alive, to be a living, contributing cell. And your cell is in interphase for about 90% of its life. And then for a very small amount of time, only about 10%, we will actually go through division. So all of this is interphase. All of the living and growing and copying and all of that, that's interphase. But then actual making chromosomes, doubling those chromosomes, splitting the chromosomes apart, making new cells, that's mitosis. So in the grand scheme of things, we don't spend a lot of time dividing. We spend a lot of time living in terms of a cell. So pretty much we're getting ready to divide, and now we divide. That's kind of how it's split up. So if we go back to that slide that I skipped ahead from. Okay, sorry, it won't let me do that. Anyway, I'll show you that slide at a later date, and I can show you what interphase at that level looks like, and then what mitosis at that level looks like. So all cells have a life cycle, air quotation marks, where it starts from the moment your cell is formed. So your cell is formed from mitotic division, and then it chooses one of two pathways. Either the cell grows and matures and it divides again, or the cell grows and matures, but it never divides again. Very few cells choose this pathway. And I say choose like they have conscious thought in it. They don't. Mostly your brain cells, excuse me, are going to be the ones that do this kind of life cycle. Your brain cells, your nerve cells, and your muscle cells. They will grow and they will mature, but they don't really divide. Whereas nearly every other cell in your body is going to go this pathway. They will grow and mature and divide. And that would be things like your skin cells, your blood cells, nearly every other cell you can think of, even stem cells. Liver cells are a little bit special. They can start off this way and end up going this way if they need to regenerate or do something else. So we can take our two sections, our interphase and our mitosis. So let's work with interphase alone. We can split interphase up into three sections: G1, S, and G2. The G stands for gap, but other books and other people also refer to G as growth. So I want you to know both ways. So it really stands for gap, but people also talk about it in terms of growth. Now, your regular cell cycle is going to be made up of G1 to S to G2 to M. So you start off here, you're formed after mitosis, you go through G1, you then go through S, you then go through G2, you divide again, you make some new cells, and the whole process repeats itself. That's the cells that go through active division. The cells that don't ever divide again, the ones that don't go through active division, they're formed during mitosis, they go through G1, and after G1, they go to another state that's called G0, or resting phase. So they completely skip S and G2 and all that. They never go back to mitosis, unless they're liver cells, and they need to. So interphase is divided into three phases: G1, or first gap, and in first gap, you're just living, growing, getting bigger, doing your everyday thing. After G1 comes S. S stands for synthesis, so we're making something. Specifically, what we're making is DNA. We're going to copy our chromosomes, because don't forget, this whole life cycle is about preparing us to get ready to divide, to go through mitosis. So we have to copy those chromosomes. After we've copied our chromosomes, we go through G2, which is second gap, or second growth. And in this phase, the cell is literally preparing itself for division. We get even bigger. We start copying the organelles that we have, the proteins that we have, the enzymes that we have, the membranes that we have. Let's go ahead and add I know we know proteins are enzymes, or enzymes are proteins, but let's add enzymes here as well. Excuse me. Okay, so that's pretty much interphase. G1, S, G2. In G1, we're growing. In S, we're copying chromosomes. In G2, we are growing some more, and we are producing double the amount of everything that we have inside of us in preparation to divide. So if we look at what's taking place in our nucleus during interphase, remember interphase is G1, S, and G2. If we could stain a cell that's in interphase, you'd see a nucleus that's well defined. You'd see lots of DNA, but it's all strung around in the cell. Like I took a huge ball of knitting yarn, I unwound everything, and I just threw it out there. Here you go. Figure it out. There are no actual chromosomes. So when DNA is loosely packaged in these long, thin fibers, it's called chromatin. So in an effort to prepare for mitosis, one of the things that we will do is we're going to replicate into chromosomes. We're going to replicate our chromosomes. We're actually going to form chromosomes, and we're going to make copies of all of our DNA. Now, in eukaryotic DNA, when we talk about chromosomes, we're talking about some mostly DNA and also some proteins. Those proteins are called histones. Now, once we've copied our DNA, we're also going to go ahead and copy everything else inside of our cell. All of the cytoplasm, all of the organelles, all of the proteins and the enzymes, all of the membranes, because this cell is going to split itself into two to make two new cells. You have to make sure that you have enough of everything so both cells can survive and can grow and continue the process. Now, S phase is all about copying and replication, and hopefully you did the replication video before you did this video, because if you don't, now you're probably confused. So go back and look at the replication video again if you need to. So in S phase, that does happen during interphase. We are going to replicate or copy our DNA. So in synthesis, we're going to take the existing DNA that we have, and we're going to make copies of it so that we have enough for two cells. Not two new cells, but two cells. A human cell is going to duplicate approximately 3 meters of DNA. That's a lot of DNA, just to give you a frame of reference. Think of a meter stick. Now think of 3 of them put together. That's how much DNA is in one little tiny cheek cell, one little tiny, epithelial cell. Every cell that's about to do this has about that much DNA in it. We have to copy everything because your DNA is your little instruction manual. A cell needs all of the instructions. We have to make sure that each cell is going to get not just a complete set, but a complete and identical copy of what the original cell had. The error rate, like we've discussed before, is about approximately 1 mistake for every 100 million bases. Excuse me. We have about 3 billion base pairs, so about 6 billion bases in the mammalian genome. Anything that's a mammal, you're looking at about 6 billion bases, or 3 billion base pairs. So that equals out to be about 30 mistakes for every cell cycle. These mistakes are mutations, and they're mutations specifically to your somatic cells or your body cells. These do not affect your gametic cells. We're not affecting our sex cells in any shape and form. So that's why if I have cancer, if I have skin cancer, I could give birth to a perfectly healthy, happy, very cancer-free baby, because the cancer that affects me is in my body cells, not in the cells that gave my child life. So, 3 meters of DNA. That's a lot of DNA. So, how are we going to arrange this? Well, DNA is organized into chromosomes when we're getting ready for division. It just reduces on the chances of making a mistake or breaking or leaving out a piece accidentally. So what we do is we take our double helix. So if you follow along with me on this part of the screen. This is the picture that's going to go with these words. So you can write these words down. I mean, you should be writing these words down. But here is the picture that's going to help you understand what these words mean. So I'm going to give you like a second or two to get some writing down, and then I'll go through this picture with you. I'll wait. Don't take your time, though. Write fast. All right, so we start off as just bases. A, T, C, G, A, T, C, G, 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 C, A, T, whatever. Just those 4 nitrogen bases. Those bases are attached to a sugar and a phosphate, and that makes a nucleotide. DNA is made up of a bunch of nucleotides, 2 sets because we have 2 strands. So we wind ourselves up into this helical arrangement that we know as DNA. Each piece of DNA is then wound around individual reels, let's call them, almost like reels of thread. But these reels are actually proteins that are called histone proteins. We take bunches of histone proteins and we can then stack them together. Once we've stacked them, we can then bend them around themselves to make like a loop-like thread. Then we can take all of these loop-like threads and lay them one across each other, and eventually we can build a chromosome from this. In class, I am going to try to have a model to show how this works. So let's go through the words now. So we know that DNA is organized into chromosomes. We know we have a DNA double helix that's wrapped around histone proteins. And I always think of it as we're taking bunches of thread reels and wrapping pieces of thread around all of the reels. Or some people describe it as pearls on a string. Same concept. Once we have them wrapped around, we now have a complex. It's a complex between, or an association between, DNA and proteins. This association is called a chromatin. So when you have the proteins and the DNA around each other, like this, it is a piece of chromatin. We can then take that chromatin and arrange it into long fibers. And this is the format that DNA exists in when it's not dividing. Okay, it's just strung out wherever in the cell. But when we do need to get everything all nice and neatly packaged, we can take those fibers and we can condense them, bend them and fold them and squish them together until they take on the appearance of that like fuzzy H or fluffy butterfly or whatever you want to think of it that we know as a chromosome. So after DNA is duplicated, we're going to condense the chromosome. This is another picture that pretty much shows what the last one shows. So here's our double helix. We're going to take our double helix and we're going to wind it together over these histone proteins. Then we're going to take those proteins and we're going to wind them into a thread. Then we're going to take the thread and wind the thread on itself. And then we're going to take that thread that bigger thread, the chromatin that we found, and loop it so that we have a chromosome. Okay, so this is where it starts to get confusing. So I'm going to try to go slow, and I need you to really focus and really pay attention. So get rid of the distractions. When we reproduce or replicate or copy a chromosome, we are duplicating that chromosome. So each part of the chromosome is what we call a chromatid. So just see if I can. Okay, so where I've just scribbled in black and red, that is one chromatid. And now in blue. Okay. And then on the other side, we have another chromatid. So we have 2 chromatids making up a chromosome. Okay? And these are called sister chromatids because they are pretty much identical to each other. They contain identical bits of copied information. Okay, so we start off as a chromosome, but we're single. It's just one section. Okay? Those chromosomes can then replicate themselves or copy themselves, and they will make two sets or two pieces, which are known as sister chromatids. Okay? So each chromosome is made up of two sister chromatids, also known as double-stranded chromatids. Same thing. So let's try it again. Each chromosome is made up of two fluffy sections. I always refer to them as arms and legs. So each chromosome has two sets of arms, which would be on top, and two sets of legs, or two arms, which would be on top, and two legs, which would be on the bottom. By themselves, so one arm and one leg, sorry, I'm touching the screen like you can see me. So one arm and one leg would make one chromosome. When they replicate, they make another arm and another leg. Together, it is known as a chromosome that's made up of chromatids. They are identical because they contain the same amounts and same copies of the original DNA material. Where they meet and where they're held together in the middle is called the centromere. So when these chromosomes split apart, like when we're dividing them, they release each other at the centromere, and now we have a chromosome here and a chromosome there. This is called a separation of the chromatids. We'll go through it again, I promise. Okay, mitosis. So in mitosis, we are dividing our DNA between two daughter nuclei. We're taking all of the DNA in the nucleus of one cell. We're going to make enough copies so that when we split into two separate cells with two separate nuclei, we can have two sets of identical DNA. Okay, it happens in 4 phases. So now we're at that part of the cell cycle where we're only in that section for about 10% of our life. It's split into 4 phases. And collectively, they're called mitosis, but individually, the phases are known as prophase, metaphase, anaphase, and telophase, or PMAT. Okay, there should be a video here. I will play this video in class. Now, all the video shows, is how the parts and pieces move. Once we have moved all of our DNA around, and we now have enough copies of everything, the last thing that's going to happen is the cell itself has to split in half. That process, that splitting of the cell, is known as cytokinesis. And it happens in 2 different ways, depending on if we are talking about an animal cell or if we are talking about a plant cell. Animal cells make a feature called a cleavage furrow. So this right here would be the cleavage furrow. What it really is a series of actin microfilaments, which are part of your cytoskeleton, that form a circle all the way around the cell, and then they start to constrict. They start to pull themselves closer and tighter to each other. So a furrow forms, and that furrow moves all the way through the cell, splitting the cell in half, or well, in 2. Okay, so it's almost like you took, Play-Doh and you tied string around it and you pulled the string all the way through, and now you have 2 pieces of Play-Doh. Same thing that's happening to your cell. Okay, so this was one cell. Here the actin fibers are condensing and constricting and drawing and pulling that cell apart until now we have 2 daughter cells. Okay, so this is what it looks like in real cells. These cells are in interphase. You can't see any chromosomes. All you see is it just looks like a bunch of threads, like everything's just kind of wherever. This is prophase. In prophase, for the first time, you can actually see chromosomes. So see here, this is your nucleus. Let me define this a little bit for you. This right here is the nucleus. See how on the inside you can't see any actual chromosomes or anything that looks like chromosomes. Now that we're in prophase, this is the nucleus. We can actually see some kind of shape. So chromosomes form during prophase. Okay, and then some parts start to move away from each other. In metaphase, now the nucleus at this stage has kind of broken away, but I'm just going to circle where you'll see the chromosomes. So that's what you're looking at. In metaphase, the chromosomes line up in the middle. Okay, then after metaphase, I'm going to move this slide up a little bit, guys. Okay, after metaphase comes anaphase. And in anaphase, the chromosomes start moving away from each other. So again, this is where you should be looking. Okay, now in telophase, in this picture you can see it much better. I would actually call this late anaphase. But see, look, you have a bunch of chromosomes over here and a bunch of chromosomes over there. So in early telophase, we're going to start forming that band of microfilaments. If you look closely, you can kind of see it starting to form, like right here. All these lines that are coming together, that is your band of microfilaments. Okay, so the chromosomes oh boy, how do I say this easily? The chromosomes are on two sides of the cell. Okay, and then we start to form our filaments. Then in late telophase, you can actually really see the furrow, and you can see those chromosomes start to lengthen. So, furrow and chromosomes lengthen. Okay, and those are all the parts of mitosis. If we look at it again, this is it in animal cells. This is a stained version, but it shows pretty much the same thing. Here is interphase, you can't see any chromosomes. Here's prophase, you can start to see the formation of chromosomes. In metaphase, they start to line up along the middle of the cell. So that's what's happening here. Here is like they're moving to the middle, and here they're at the middle. In anaphase, they start to move away from each other. And in telophase, we've made that cleavage furrow, and here are our chromosomes. To give you some perspective, everything in blue is DNA. So the blue is DNA, and the green is the cytoskeleton. Just to give you some perspective. And then the red is just the cytoplasm. Okay, so you can kind of see how everything's happening there. No, oh, wrong button. Okay, now in plant cells, it happens a little bit differently. Instead of making a band of microfilaments, we're going to form, or instead of making a cleavage furrow, we're going to form a cell plate. So in an animal cell, we make a cleavage furrow, but in plants, we make a cell plate. Instead of that plate being made up by microfilaments, this plate is made up by vesicles. So a bunch of vesicles are going to start moving into the middle of this whole big new cell that we've made, or this whole big stretched out cell that we've now made. Vesicles are going to move to the middle, and they're going to line up in the middle. These vesicles are coming from the Golgi apparatus. The vesicles are then going to fuse to each other. And when they fuse to each other, they form two cell membranes, one on the left and one on the right. So pretty much what they will do is they're going to come and put a new membrane right in the middle of the cell, and then they're going to lay cell wall down between the membranes, and that's going to separate the cell into two separate plant cells. So if you look at the picture, this is what it looks like in real life, like all these little dots, those are the vesicles, but here's the cartoon version, which is easier to understand. So here we have our plant cell. We already have our DNA on the left and on the right, so these vesicles from the Golgi apparatus are just going to come and lay themselves down in the middle. Then each vesicle is going to fuse to the one above it and the one below it until we have like one huge vesicle. That is our cell plate. Now, on either side, on this side, we have a cell membrane, and on that side, we have a cell membrane. Once it runs the length of the cell, we're then going to come and lay down cell wall in between the membranes, effectively splitting this plant cell into two daughter cells. All right, here's a close-up. So here are your vesicles. All right, just more pictures. The more of these you can see, the better the better you can have a visual to go with what I'm talking about. So this is what the cell looks like in interphase, nothing special. Prophase, we can now see some chromosomes beginning to form. Metaphase, everyone's lined up in the middle. Anaphase, they're moving away from each other. Telophase, the chromosomes are effectively on the left and on the right. They've recondensed, so now you can't really see chromosomes anymore, and now you have something happening in the middle to split your cells apart. In this case, because it's a plant cell, we are forming a cell plate, and then we're forming cell membrane, and then we're laying down cell wall. All right, another picture, just bigger. Okay, so where did mitosis come from? Well, mitosis in eukaryotes started off or first evolved as binary fission in bacteria because pretty much works the same way. So in a bacterial cell, remember their chromosome is just one large circular chromosome. And remember, they don't have any membrane-bound organelles that they're going to have to reproduce, but everything else is pretty much the same. So in a bacterial cell, we start off at one point within our chromosome, which is called the origin of replication, and we separate the DNA, we make all the copies that we need to. Once the copies have been made, the DNA moves to either side of the cell, the left and the right. The cell is going to elongate to accommodate that. We're going to have a ring of proteins that form in the middle, and the cell is going to pinch off into two separate cells. Pretty much mitosis. Okay. Now, a lot of prokaryotes and simpler organisms utilize something very similar to mitosis to divide. You see a lot of similarities between the two. So this is how protists do it. And this is a type of protist called a dinoflagellate. This is another type of protist called a diatom. But here, look at the very first type of eukaryotic cell, yeast. Simple, but it does it the exact same way. Here, we're making the cell bigger. We are moving apart the chromosomes that we've replicated. We're going to split the cell apart in the center. And these are how eukaryotic cells do it. So over time, the process itself hasn't really changed majorly. We've just fine-tuned the little pieces that we need to make those particular organisms successful. Okay, so hopefully this was easy to understand and not too much trouble. Again, we'll go over it in depth in class, and I'll see you guys soon.